So talking about muscles, I think it's cool. We're going to talk about Grasshopper in a second. So Grasshopper has three pair of appendages, and as we all know, those hind legs, good for jumping. And let's see. So we have the tibia below the joint, tibia, IBIA. And then below the joint we have the femur, which is, we have a similar naming with tibula. We have the fibula too, but they have the tibia, we have the femur, where the large muscles in the grasshopper. You don't think of grasshoppers, have muscles? They do. Insects have those muscles. And when, to get ready to jump, flexor muscles have to pull the tibia toward the femur into a Z shape, and then extensor muscles contract and pull that tibia into a straight line really quickly, and that gives you that powerful force that makes that grasshopper jump. Okay, so forget I misspelled this OR on that one. And so draw a little diagram. We'll say this is the femur, this is the tibia, this is the femur. Let me go extensor muscles kind of crowd around the middle, and then to flex it, we'll make those purple. So the flexors are out here. Flexor, and this is extensor. Okay, so to jump, these flexor muscles are going to pull the tibia. If I should use, if I can erase that. The flexor is going to flex, it's going to bring this tibia in, so it's going to be more like a shape like this. And then all of a sudden the extensors pull down and will then kick that tibia up to have that grasshopper jump real high. I don't know if you guys used to catch grasshoppers. I used to when I was a kid. Hopefully I was nice to them. I never hurt animals or mess with insects. I'm sure I didn't, but used to catch them, put them in a jar, check them out. Pretty cool. But we need to make sure we ethically treat animals, even if we're interested in them. So a place where bones meet is called a joint. Let me get the red back. So joint. And moving joints is called articulation. Articulation. So you articulate your joints when you walk. Hold your pencil. What I'm doing right now with my little sketch pad here. And cartilage is tough, smooth tissue that covers the bones where they meet, prevents friction. And also you have those bones kind of pounding against each other. So besides the friction, it's also they can shock each other, impact each other. If that happens too hard, you can break the bone. So we want to have those absorb some shock as well. And you have this stuff called synovial fluid to lubricate the joint. You want it nice and lubricated. When you lose that lubrication, that's when you have things like arthritis happen. And if you know somebody with arthritis, it's not fun. I have a family history. Hopefully I'll be good. But the joint capsule is the covering of the joint. And you might have heard these words, tendons and ligaments. Need to know the difference. Tendons attach muscle to bones, and ligaments attach bones to other bones. I can't go behind my little image there. So you might need to label something like this. This could be on the exam. So you have to be familiar with it. So right here we have the bicep, the human elbow. So make sure you review this particularly. Attaching muscle to bone, that is going to be a tendon. And back here behind the bicep, you got that tricep. And this structure here connecting this tricep to the bone, that's going to be a tendon. And right here we attach the humerus to the radius. That's going to be a ligament. And behind here, let me just remind myself, what is back there? Okay, bursa. Just a sac with some liquid in it. Okay, helps to feed liquid into that joint to keep it lubricated. Another diagram you want to be real familiar with. Another slightly different angle, but attaching the bone to the muscle, that has to be a tendon. And of course this muscle in front is the bicep. The bone that the bicep attaches to is the radius. I-U-S. And the other bone is the ulna. So 2 bones in your forearm allow you to rotate your wrist around. And back here, tricep. And the bone here, humerus. And you heard the funny bone. You've hit your funny bone. You hit a nerve right here. You feel this tingling up your arm. So I don't know if that's related or not. So you need to be able to draw a diagram of a sarcomere showing the actin or the myosin filaments, the actin fibers, how that's structured. M line is in the middle, Z line is on the side. So let's get to it. So basically you want to be able to draw something like this. So here we have our actin. We have our thicker myosin. Let me drop a new color on you to help with that. Myosin. See, it's thicker. It's thicker because I'm drawing it thicker, but it really is thicker. So myosin. These right here, actin. This right here is going to be the M line. This right here is the Z line. And on, what just happened? Huh? Ooh, it's okay? All right. I pushed a button, something happened. I was freaked out, but okay. Yeah, and on these, this is where, let me get another color involved here. Let's get some yellow. This is where you have the myosin heads. Boom. And they interact with that actin. Ah, I like those colors. That's nice. Hopefully you use some colors on yours. That does make, I swear that makes a difference. That looks nice. Man, that's nice. So yeah, let me label that. Let me color code this. This is the myosin heads. And that's so important. Like we talked about before, this is where you have this interaction. Calcium comes in here, allows the head to grab the actin and push it this way. And just like just somebody's rowing a boat. If you ever rowed a boat, boom, boom. It's just the oars are like these actin heads, just pushing back, propelling that boat forward. Now, this would be, I would say, this would be relaxed. When it's flexed, you see this area here. In a microscope, it looks clear or light. This will get much shorter. It might just be this big, and that's when it's flexed. We'll get to that, I think, in our next soon here on the slides. We'll go back to red. In a relaxed sarcomere, the Z lines are further apart, so they're like this, and then boom, they're closer when it gets contracted, when it's flexed. The light bands are wide. You see a lot of emptiness in there, and you see that on the test you might have a electron micrograph. In the middle, you got that M line, and again, you're gonna see a little more visible light on either side of that M line that I can represent with this white right here. Oh yeah. So it's like this when it's relaxed. When it's flexed, this white area is just going to get a lot smaller around the middle. So this is relaxed. This is contracted or flexed. Okay? And let's go, oh yeah. So you see that, the M line in the middle, the thick, this is myosin. See a better color, myosin. Is this, the dark band, and the actin is surrounding it. But yeah, fully relaxed, contracted. You'll see this is the distance between the sarcomere, and it's wider. This is shorter, the shorter gap between the Z lines. You also see this I band here. What do you think happens to this I band, this white space around the Z line? When you flex the muscle, it gets smaller. So let's check this out. Let me get back to some red. M line, boom, right here. Let's point right to that, right in the middle. Z line, boom, right there. That's just one example. Dark band, basically right here, seeing that myosin. Now remember, there is actin in there, but mostly myosin. Light band right here. Maybe the I band might show up, and the sarcomere is this whole unit right there. All right, very good.